It starts with you, locally, in your lab, in your community, in the moment you move from possibility to progress. With Thermo Fisher Scientific, those moments don't stay small. They scale, they accelerate, they turn insight into impact. In structural biology, in biopharma, in clinical research, taking shape through translational omics, unfolding in proteomics, decoded in metabolomics, protecting our world through environmental food safety. What begins with you doesn't stay small, it grows. From streets to cities to continents, expanding, accelerating, connecting, until it becomes something greater. Global impact. Impact that reaches beyond today with next generation solutions to go beyond today's performance and beyond our generation. Fueled by innovation and built for purpose. Enabling our customers to make the world healthier, cleaner, and safer. From local insight to global impact. This is where it all comes together. Welcome to the 2026 Thermo Fisher Scientific Users Meeting.
And now, please welcome to the stage Thermo Fisher Scientific President and Chief Operating Officer, Gianluca Pettiti.
Well, good morning to you all. This is my favorite meeting of the year, and apparently not only my favorite because we have a lot of people standing this morning, so thank you all for joining, and apologize for my rocky voice. You're going to have to blame either my AI coach that says that a rocky voice is a lot more charming, or perhaps the fact that if you live in Boston, this is the first opportunity we had in the year to come and defrost in San Diego, because I tell you the weather there has been pretty miserable. Well, I have to say we're incredibly excited for what we have in the show this year. And we're excited to be here with all of you because really this meeting, it's about you, about the impact that you have through the technology and the solution that we have the honor to actually bring to you every day. And that's incredibly important because you have always been at the center of what we do at Thermo Fisher Scientific, as our mission is truly enabling our customers, enabling you, enabling our partners and collaborators across the globe to make the world healthier, cleaner, and safer. And there's never been a better time in science to do that, because the innovation and the technologies that you have at your disposal are truly transformational, and we're very excited to talk about that. We're also excited because at the company we have the opportunity to do that at scale. In fact, we have 125,000 colleagues that are coming to work every day to support you, to support our customers, and through you and our customers and partners, ultimately to support hundreds of thousands of patients that are in need of help. We invest at scale. We invest more than $1.4 billion in R&D every year. We have incredible scientists. Many of these are in this room that, again, are working with you day in and day out, and we get the benefit of scale as well in our revenue. But scale is really not what matters the most. It's truly the proximity with all of you, is the fact that those thousands of colleagues can spend time with you and with our customers every day. That's where the unlock happens. And this year, similarly to last year, we're equally excited about what is happening in proteomics. In fact, you have seen just over the last 12 months the progress that have been made with some of the technologies that we showcased last year at ASMS, with a combination of techniques that spans from mass spectrometry to structural biology to high affinity proteomics now coming together to actually help you all to go and find the secrets that are still hidden in the complexity of a science that have kept all of us connected with it for so many decades. And we're just getting started. We're just getting started, but to actually advance even faster, we need to continue to have the best tools, the best technology, the best partnership. And at the company, we worked over decades to assemble what today is a world-leading portfolio in solutions for proteomics. They were designed, developed, and brought to you all with the perspective of simplifying the way you work, giving you a lot more in terms of detection capabilities. And now we're enhancing those capabilities with novel software solutions, including artificial intelligence. So we're incredibly excited about it. And in the middle of the page, a lot of what you hear at ASMS is about detection technologies. And I have to say, this year we're bringing it to a whole new level with two new transformative technologies that are actually going to allow us and you and the world of science to go even deeper, to start to look at proteoforms and to understand biology in a level of depth and specificity that was not thinkable just a few years back. The structure, the function, the connectivity between everything that you study and disease progression, this is going to be truly transformative. So I can't wait. And you have seen us as well being active in the space of software and digital solutions with a couple of acquisitions this year with Proteination and MS8 that will complement our solutions from a hardware standpoint, complementing the ecosystem of solutions that you have available every day in your laboratories to advance science. And that's, again, incredibly exciting. But don't take my words for granted. Actually, we see that in the energy and momentum that we are all experiencing in the space of proteomics. And in this page, you have an example on how our gold standard technology for proteomics detection with our Orbitrap Astral Zoom that was launched, as you know, at this conference, and that has been the most successful high-end mass spectrometer ever launched, not by our words or our definition, but by the publication that you all have produced over the last few years. And for that, we're incredibly thankful for your role in advancing the world of science and technology and innovation. You've done incredible work with the technology. And the combination of mass spectrometry and high affinity proteomics with the Olink technology is coming together now in large cohort study, in large population studies, where countries are relying on our solutions to actually advance one of the major health study across the globe, like the UK Biobank, as well as the PRECISE program in Singapore, that now is the first of its kind combining mass spec and Olink. So it's incredibly exciting to see not only that those technologies are used for discovery, because they're truly the best at supporting you in what you do every day in a lab, but they're also scaling. And so that's the essence of what we call from local insights to global impact. It is truly global and with an unprecedented scale. And so I truly can't wait to see those insights that are being generated from these large population studies and how they're going to transform not only the way that we prevent and diagnose disease, but how they're going to catalyze and inform novel drug target and therapies. I think we're truly at the verge of something special, and I'm so glad that we're in this all together. And as you know, at the company, we're not only about what is one of our crown jewel, is our chromatography and mass spectrometry franchise, but we try to help customers, and in this slide you have an example on our biotech and pharma customers and offering from discovery to getting their drugs to market. Everything from core research to actually supporting our customers with process development for their therapies, all the way with clinical trial, with our clinical research group, to potentially manufacture drug for them with our CDMO business. So that's an exciting value proposition that ultimately connect those insights that you're working incredibly hard to produce every day to the action at a global scale through diagnostics and therapy. And so it's an incredible platform, and you should, again, all feel incredibly proud of the role that you play in it. And if that's not enough, what about artificial intelligence? Artificial intelligence has the potential to enhance everything we do. The way we work, the way we live, I can tell you for sure the way my kids live. I have a 10 and a 12 years old, and they're already playing with AI every day. That's quite scary. But that's transformative. And the benefit that we are going to see will be quite remarkable. And you know, there's hype, there's practical example on how artificial intelligence is truly transforming the way we work. And then I think there's a lot of potential in the future. And you know, typically we're quite humble as we think at innovation. We know where we're good and we know where we need help. And so over the last year, we established partnership with very strong leaders in the artificial intelligence space. OpenAI supporting us across the company with a frontier partnership, and we focus that effort on clinical trials. Imagine the amount of data that we have as a CRO with tens of thousands of clinical trials data that we can help our customers with. A very strong partnership with NVIDIA that will produce and is producing already the first autonomous set of instruments that will come to market in the second half of the year. So that's incredibly exciting. And the partnership with BenchSci and TetraScience, they're focused on data, because we can't be naive. In the world of science and laboratories, data, it's a bottleneck. And so resolving that aspect to take full advantage of artificial intelligence is incredibly important. And so we're working very hard at it. So we're excited. And now, you know, it's always challenging to come and talk in front of a room full of incredible scientists and researchers and collaborators. And so I put here my piece of science on artificial intelligence, because being an engineer by background, I spend a lot of time in trying to resolve math problems. And is a news from yesterday that a model from OpenAI was actually able to resolve a problem that has been unresolved for more than 80 years. A mathematician called Erdős from Hungary. It was a genius. Probably some of you who is familiar with Erdős in the room? Great, few of you. Well, anyway, he's a famous European mathematician, and he left behind very complex mathematical problems. And for the first time, an AI model was able to resolve one of these problems. Is a planary dot problem. I won't get into it. But no one resolved it for 80 years, and OpenAI was able to do it. So we're starting to see actually a little bit of creativity in the artificial intelligence models, and so that's exciting for science as well. And we believe that this is going to catalyze a lot more data generation. You actually need to make sure that your laboratories are up to speed with the right instrumentation, with the right connectivity. So our team is here to support you. If you need a laboratory refresh package, we're happy to support you with that. So with that, let me close and start with, again, a thank you to all of you. We wouldn't be here. Thermo Fisher Scientific and the mass spec business wouldn't exist without your support, your leadership, your contribution. Secondly, we're living an incredibly exciting time in science. And that's exciting because of the work that you do again. This conference is about you, about your work, and the incredible things that you are producing with quite good technology that we are producing. So we're proud partners in that. The future is even brighter, and it needs to go through a scaling process. We're a great partner for that. So let's continue to find local insights and then bring it to the globe. I was in Latin America last week, and I tell you the need of innovation they have down in Brazil is incredibly important. So that's a great example. So from local insight to global impact. And let's take full advantage of artificial intelligence. We have great partnership. We're producing great solutions. We want partnership with you as well to make AI matter in what you do every day. So with that, I'll hand it over to the rest of the team to continue the keynotes for the morning. I couldn't be more excited to be here on behalf of our company leadership team and our analytical instruments group. I thank you for your presence, and I wish to you all an incredible 2026 ASMS. Thank you very much.
Please welcome to the stage Thermo Fisher Scientific Vice President of Innovation and Research and Development Strategy, Ian Milcrest.
Reflecting on your AI comments, I think next year will be digital twins or avatars up here, right? We'll do it that way. So it's always a pleasure to introduce our new technology. You'll stand in room as people stood outside as well. I was just reflecting a little on some of the advances in proteomics. I remember when we bought Life Technologies back in 2014, the COO at that time, because they're all genomics, he pulled me aside and he said, you know, he said, Ian, proteomics is going to have its day in the sun one day. I said, all right. We will, and we are. And I think that's where we are today relative to the technologies and the work you're doing, whether it's through scale proteomics or through structural biology, the technology we have, and what you're doing is really pushing the boundaries of science. So, you know, our day in the sun is here, and I think it's reflected in some of the comments that Gianluca made about scale. If you think about biobanks, we were never invited to biobanks 4 or 5 years ago. Now we have the precision, we have the depth, and the speed of analysis to be able to do those. So it's really, really changing. And when you combine our technologies with Olink and mass spectrometry, cryo-electron microscopy and mass spectrometry, bringing these hybrid technologies together, really game-changing in terms of what we do as Thermo Fisher and providing solutions for you. So just for us, this is important. Obviously not as important as the papers you produce, but the industry does acknowledge the technologies that we produce. So a number of acknowledgements here, R&D 100 for the Stellar in terms of its technology, our HPLC technology in terms of sustainability by the Scientist's Choice. The Astral last year got awarded the Horizon Prize from the Royal Society of Chemistry. And the Astral Zoom was one of the top 10 innovations from analytical scientists. So we're proud of that, and we realize the impact. But the impact, as Gianluca just said, actually, is more about this. right? It's about what you do. And if you think about 3 years ago since we introduced the Astral, and you all started getting those, there's been over 1,100 articles published on that platform between the Zoom, and probably got more than that now because they're produced at a rapid rate. I think what's stunning, though, is how many fields of research that's covering, right? Proteomics and structural biology and everything we do in omics is not one thing. That's almost 400 fields of research, which is quite stunning, actually. And I think what's more important, there has been an inflection point. If you actually look at our publications over the years, and it's about 150,000 Orbitrap-related publications, there's this massive inflection in 2024, and all these papers are getting published in half the time. So we're proud of that, right? We're able to do more science faster and make more breakthroughs, and whether it's determining diseases or whether it's developing therapies, this is the impact of the science. So we thank you for the work you're doing on these platforms. And our goal is to keep developing these platforms so you can do this great science. New instruments this year, very, very excited to introduce 4 new hardware platforms, some new separations technology, a lot of software. And we talk about software a lot. It's a massive investment for Thermo Fisher, both in artificial intelligence, machine learning, but also our core software. So introducing this year is the new hybrid technology. It's the most powerful tribrid technology we've ever produced, the Apex. So we'll talk a little bit about that and the advancements we've made in that platform. That's our most powerful research tool that we have in our portfolio. New hybrid technology with the Excidion, which is really tailored towards biopharmaceutical analysis, both structure and also pharmacokinetics. We have a new HPLC, which is an inert path HPLC system, which we've never had before, which is extremely important, particularly for sticky molecules. You start thinking about oligonucleotides and some proteins which get stuck on metal surfaces. And we have also partner columns to go along with that for oligonucleotides, these new monodispersed columns that we've put in place, SURE packs, for both oligos and proteins. We also have a new GC Orbitrap. Don't forget, we still do GCMS. A lot of people do GCMS, particularly for POPs and dioxins, etc. So we have a new GC Orbitrap with new sensitivity, new capability, new resolution. Like I say, in 6 new software packages as well, along with the programs that we've acquired through Proteinaceous and MSA. And we're very excited to bring those technologies on board. So where we accelerate your research, and I'm going to go through these technologies a bit more depth in terms of where we accelerate research. Proteomics, like we said, is a huge, huge field right now. How many application spaces that covers? Several hundred. Metabolomics, translational, and structural biology are all really tied up. And what the Apex brings, and our tribrids have always brought for years and years, is a lot of experiment flexibility in terms of what they can do, in terms of how they fragment, and in terms of how they detect, how we move ions around in that technology to bring new workflows, one from the front end in terms of how we put samples into the systems, but also how we move ions around inside those instruments to actually do very, very clever experimentation. And we keep getting feedback on, I wish you could do this experiment with this new modality, and that's what really drives us to develop new technology. So here's the rise of molecular challenges of tomorrow, and I would say it's also today. There's plenty of challenges today that are still out there that we need to resolve. So this is the Apex. It is a family of products, and I'll describe that later on. As we did with the Orbitrap Tribrid Ascend, we have different models for different application spaces just because of the breadth of capabilities. So it's one platform, multiple workflows, right? It supports multiomics, it supports biopharmaceutical capabilities, structural biology, but also small molecules. For those of you who've been around, you know that we also have a tailored system for small molecules. The IDX and the IQX have been around for quite a while, and they're very much structured for small molecules, and we're going to focus. But it's all based on workflow at the end of the day, is how do we separate, and how do we do analysis, and then how do we actually do the data. And you can see the investment that we make in software. I think very importantly is 2 new ones there, Proteoform Studio, which came out of the Proteination acquisition, and ProCyte Native. A lot more work going on around proteoforms, which has always been a massive challenge in science, right? Isoforms and proteoforms are a massive challenge, whether it's in understanding disease or whether it's developing drugs and targets. And also a lot of more work going on in the native state now. Capabilities in our systems to look at native molecules is becoming more and more profound. And I think we have tools now, both on the instrumentation side and also the software side, to address those challenges. For those of you who've been around, many of you know the tribrids well. We actually launched our first tribrid in 2013. It was the Fusion. I think we spent more time actually arguing about the name of the system versus the geometry of the system. But it's very unique, obviously, with quadrupoles, Orbitraps, and linear ion traps. And we can put those together in any way we want and move ions around. It allows us to do very, very elegant experiments. And those have evolved over the last 12, 13 years into terms of how we actually make those perform and the performance. And I'll show you what we've done with those technologies. It's always been a system that's had multiple fragmentation modes, so you can take apart any molecule you want in any different way, whether it's with HCD, CID, we've added ETD, PTCR over the years, we've added photon dissociation, and also multiples of those too, right? So we see HCETD, we see different fragmentation modes you can pull together to actually take molecules apart in different ways. This is also a very intelligent system. As you well know, a lot of you have these tribrids. We can move ions around. We can do real-time search. It's been a powerful tool for tandem mass technology in terms of doing TMT quantitation. It is the TMT machine out there for doing quantitation, supporting our 32, 35-plex TMT. AcquireX was an intelligent acquisition for small molecules. A lot of method templates. So it's a very, very powerful platform that we continue to build on in terms of experiment flexibility. So what have we done now on the Apex? So we've added some new technologies to move it up even to the next level, significant performance on the Apex. So we've added an IR laser to the system. This gives a new orthogonal fragmentation capability. It also gives gentle desolvation. I'll show an example of that. Many molecules hang on to detergents and water. We can actually use an IR laser to get rid of those. So it makes life a lot easier when you're doing the analysis and doing MSN. It actually improves ETD in terms of exciting ETD ions. It also is great for doing TMT. It actually improves the TMT reporter ions, gives more precision and more sensitivity. So you'll see an IR laser on the back of the system. We already have a UV laser on there on the current ascend. We've also put direct mass technology or charge detection mass spectrometry on this system. We originally introduced that on the Q Exactives, and it's been on there for a while. So in this system now, this tribrid now has direct mass technology. So we can do charge detection, and I'll show you an example of what that really means. It's very, very powerful for complex analysis, particularly with things that overlap, isotopes that overlap, large molecules where you can't actually find the charge state out. It also increases sequence coverage, and I'll show you an example of that in terms of how it helps looking at monoclonal antibodies and looking at the variable regions there. It's very, very powerful. We've extended the native mass spec capabilities, so you now have 20,000 mass rate M over Z on the Orbitrap for MSN. We've actually extended on the quadrupole there to have isolation up to 12,000, M over Z 12,000 from 8,000 previously, and narrow isolation down to 5 Thomsons. And there's no compromise on the low mass anymore with this system as well. We've also increased the scan rates on the Orbitrap to 75 Hertz. So it's gone up significantly. We've got more efficient utilization of an intelligent parallelization. There's now an optic up front that allows you to do parallelization and also MS2 before the Orbitrap. And there's some new resolution modes in there. So you can actually go to lower resolution and go faster, right? So we can go down to 1750 resolution where it was limited before. So it actually increased that scan rate. We've also gone to smart TMT acquisition capability with the end-to-end workflow. And also intelligent triggering, because you can actually put the laser and also SP3, MS3 scans together and trigger from those. And we now have an adaptive real-time library search capability in there for both small molecules and large molecules. So significant advancements in performance on this platform, both for structural biology, proteomics, small molecule metabolomics capability. We also put the new source on here. It also switches polarity 40% faster, right? So you get more scans across the peak, particularly if you're doing small molecule analysis. So significant advancements. Here's a picture of it. If you can actually read it and see it, you can see what's going on there, where we've made advancements on the front end of the source, in the quadrupole in terms of isolation, in terms of the front end routing octopole there, which allows us to do parallel acquisition. We've raised the mass range. On the ion trap, we've done some things too now. Now you've got 6 modes of fragmentation inside the ion trap, so you can go crazy there in terms of what you want to do. We've also put an oligonucleotide mode in there, which is a low-Q CID inside the trap, which allows you to look at hundromers of oligos. But more importantly, you can actually look at modifications on RNA very effectively with that fragmentation mode. And as I've already said, there's an IR laser on the background. So significant advancements in technology on this Apex platform, which will really open up new channels of information for your science. So just a couple of examples. I get to show some spectra this year, which is unusual. I usually don't. So here's using an IR laser for doing gentle desolvation. And native membrane proteins particularly hang on to detergents, and many molecules hang on to detergents. Now you can knock them off using source CID or C in the background. The problem is that they're not native anymore. They start unfolding, right? We put too much energy into the molecules. So in fact, if you use IR on the back side, you actually get rid of all those detergents, which causes that tail in the spectrum there, and actually get clear charge state and isotopes around the bottom. So very, very powerful from a desolvation perspective. A lot of people do native membrane protein complexes today. And so this is a very powerful adduct to that. Also using IR-activated ETD, and I actually stole the slide, it's an old one actually, and I should take some of the naming off there. It's actually from Nick Riley, who'll be talking later, and Josh Kuhn. But you can do IR radiation and ETD at the same time, right? So it actually enhances the fragmentation inside the trap. And one of the reasons for doing this is because sometimes with ETD you don't get any dissociation because there's not enough energy going into the precursors. This actually spins those up. So with ETD, as many of you know, you get C and Z ions, right? So you can see the spectrum before with ETD is very, very low spectral quality. If you actually put the activated photo IR in there, you actually get a series of ions that comes up. You can also put HCD on there supplementary to get B and Y ions as well. So very, very powerful capability in terms of sequencing capability for your protein experiments. We're excited about that capability as well. So for those of you who are not familiar, so we actually have direct mass technology, or charge detection mass spectrometry. We actually put this on the Q Exactive many years ago. I think one of the challenges has always been is it was relatively slow, it was relatively not very sensitive, it took a long time to do acquisitions. So one of the problems with a lot of samples, and I look at the bottom here, it says turn mess into mass, and a lot of you will see this, particularly when you have large macromolecules, or you have complexes, you have things that are heavily glycosylated, where charge states actually overlap. You can't actually measure the charge on the ions, right? So you're trying to make assertions in terms of how to correctly assign mass. So one thing you can do is actually measure the frequency, which is m over z, and the amplitude, z, of the ions at the same time. So we do that, we can look at every ion and get the exact charge state of every single ion, which then you can convert into m, right? Which you can't do on these blobs that you can see on the screen there. So this is extremely powerful technology. Now we can measure every single ion. We collect hundreds of those in parallel. There are other versions of charge detection mass spectrometry that actually measure every ion one by one by one. We do it in parallel, so we can do it extremely fast. So in this case, where you've got all these overlapping isotopes, overlapping charge states, very complex, particularly if you're looking at complexes, we can actually pull this out now and get M, which is what you're looking for. We can also actually put these things together with charge detection mass spectrometry and normal ensemble mass spectrometry and do MSN on these things to get structural information, so they both inform each other. So again, we implemented this many years ago. We've now put a new version of this, more sensitive, more dynamic range, parallel, much more faster than previous. So I'm going to give you a couple of examples, and there's going to be talks on this during the year, which is really exciting. So this is actually from Khalil. He's doing a workshop, I believe, on Wednesday morning. But you can see his mess up there on the top right. He's looking at some complexes. These are actually membrane proteins and signaling proteins at very, very low levels. This is the other problem, too. When you have very low levels, it's impossible to determine charge on the molecules, so you could never actually do a deconvolution on these things. So here's his example of his ensemble of a membrane protein complex, obviously in different forms. It can be full complex, it can be fallen apart. When you do direct mass technology on it, you can actually see how it pulls out the individual components of that complex. Then you can actually use direct mass technology to direct the MSMS and actually start looking at the individual components. So now you can see what's going on from a biology perspective. And this is going to be extremely powerful in the future, where you've got very, very complicated, very low-level samples, trying to find out what's going on from a biology perspective to understand disease and mechanism. So a nice example of really resolving complexity with the Apex. Another example from my friend Neil, who's in here this morning, looking at MABs. This is also a real issue relative to proteoforms. And I'll directly quote Neil. He said this is the most powerful proteoform sequencing system he'd ever seen. And that's what it is. So this is actually using direct mass technology on MABs, but also what the goal here is to actually sequence those variable regions, the CDRs, and there's 6 CDRs, and we'll have a talk about this later on, but you can actually successfully characterize these maps. And finding those variable regions obviously is very important from a drug development perspective in terms of what antigens they're going to bind to. So be able to actually look at these molecules now, because they're proteoforms, actually then go in and sequence those variable regions is extremely powerful and opening up new channels of information for drug development. So we're really excited about this, and I think combining direct mass technology with MSN is now going to open up a whole new channel of monoclonal antibody development in terms of how we look at those variable regions and really tailoring drug development. So very exciting. I know Neil's going to be doing a talk on this later on. So we do have tailored Orbitrap Apex models for the different applications in terms of their capabilities. So there is structural biology, and you can pick out your application here. There's not 400 of them, but there's a lot of them. Biopharma, all the different modalities that you look at, whether it's oligos, whether it's fusion proteins, whether it's bispecifics, everything else. Small molecules, like I said before, the huge push still in there in terms of metabolomics and lipidomics and the capabilities. And then multiomics, right? Obviously, a lot of people do both. So whether it's TMT or whether it's glycoproteins, etc., different models for different types of problems. And obviously, as we move forward and these new modalities come forward, we'll be developing new technologies to address those. And we know what's going on in biopharmaceuticals in terms of the new modalities that are coming through, whether it's cyclic peptides or GLPs or different things that are going on. How do you analyze those effectively? And these tools will help do that. So multiple modes of apexes here in terms of solving problems. John Lucas talked about AI and driven, and also just our software. We actually have a lot of AI-driven tools. We had them for a while, actually. MZ Cloud was actually an AI and machine learning-driven software, which we acquired several years ago. Chimeras, which is now came from MSAID, was developed, obviously, from a search engine perspective, using AI tools. We now have connectivity now into the tools from Proteinaceous, like Proteoform Studio, which is very powerful. And then the new version of Biopharma Finder, which uses machine learning tools for spectral prediction. So again, our suites of software continue to expand. We'll use machine learning and AI where appropriate to obviously give faster insights as more and more data is collected. And that's the challenge now. We're producing more and more data. How do you actually use that and find insights? So, we'll continue to do that, give a higher throughput, higher confidence, and, much better decisions and faster decisions is what we're trying to drive. So let's talk a bit about biopharma, although the Apex obviously serves the biopharmaceutical area as well, but biopharma characterization and also bioanalysis can be served by other instruments as well. So we have the Excidian Biopharma edition. So we have two versions of this. We have a regular Excidian, but this is actually the biopharma one, which really looks at mRNA and MABs and ADCs. And what we've done with this system, this is a hybrid Orbitrap technology. We put the enhanced dynamic range mode on this Orbitrap, so it's about an order of magnitude more dynamic range, which has significant impact, one in terms of what you can see, but also how much more you can see. So typically when we run this, particularly for small molecules and peptides and proteins, you can see 5 times as many precursors as you were previously, but also you'll see in a couple of spectra I show how you can actually have more confidence in some of the results you're getting. The Optimax source has got on here, so that gives us 5x in sensitivity. It does have a mass range up to 8,000, and we have new specifications on our Orbitraps at 200 parts per billion. I'll do that rather than 0.2 ppb. So that's the mass spec. And then we have an oligonuclear mode. This particular system, actually, we try to preserve your investment, can actually be upgraded to the Excidian Pro, which has a 12,000 MOV mass range as well. So once you made that investment, at least it's protected if you want to move forward. So a very powerful tool based around our hybrid technology. And again, it's an end-to-end workflow from a small molecule perspective, and we'll talk a little bit about some of these technologies like the Amplify, what we've done there in terms of developing our HPLC. It's all part of developing it. You can think about the mass spectrometry from a box perspective, but we're trying to work on solutions. Right? You've got to figure out where your bottlenecks are, and you've got to improve them. And in fact, what drives innovation is bottlenecks, because you create your own bottlenecks, and you've got to get around those. And whether it's new molecules we can't analyze, or we can't get things through fast enough, we can't feed our systems fast enough, that drives a lot of innovation. And so this is what we're doing with this particular system. So here's an example. This is actually very topical. Obviously, this is a GLP. This is semaglutide, and this is actually a metabolite to do in DMPK. And you can see on the top, with enhanced dynamic range, you actually get very, very poor isotopic data, and you're sort of making assertions there, a very poor charge state assignment. But if you put the EDR on, which gives us an order of magnitude, and so in terms of dynamic range, you start pulling these things out, which is really, really important, right, from a measurement perspective, particularly with metabolites. You want to actually know it's there and it's really there. Like I said, not only does this actually improve the data, certainly here from a charge state perspective and also an isotopic distribution perspective, but it also, you get more precursors. If you think you've got an order of magnitude dynamic range, you're going to start seeing some things that you probably hadn't seen before, and have many more precursors to do experiments on. So this is a very, very powerful system for doing that. From a workflow perspective, we talked about this, we talked about pure technology here, but also we're building workflows out. This happens to be a oligonucleotide workflow. We actually have kits now up front that do called the Oligo Select, which do the digestions all the way through. The new Vanquish system Amplify, which is very important for doing the oligos. And the reason why we developed this is because there's a lot of stickiness around oligos and proteins. So this actually is inert paths inside it, so we actually don't see any metal interaction. And the same with the columns that we developed there. And the other columns are also monodispersed columns, so they actually give you better resolution, better peak shape, and we can run faster. And obviously through to the end in terms of our software capability. So again, don't just think about the box. We have to think around it in terms of the capabilities to feed that instrumentation. Let me just talk quickly about the Amplify. This is a new Vanquish system. It does have an inert system. So you can see AMCP, which is actually the classic molecule that's used to test whether there's any metal surfaces, because it will grab onto every metal surface that's there. So in a traditional system, you'll see on the bottom trace there, that molecule just gets absorbed. It's highly acidic and it just stands. On the new inert system, it comes through, and it's used as a QC on most systems for inert analysis. So we've done that. So we're minimizing interactions, and again, very, very important for oligo analysis. This has seamless compatibility, obviously the mass spec, and obviously because we're taking metals out of the system, it also helps in the mass spectrometry side, so we don't see a lot of metal adducts as well moving forward. So, again, we think about mass spectrometry, we've got to feed the mass spectrometer with these technologies, so we're very excited with this, and also the columns that are being put in place, which now allows us to get sharper peak shape and better resolution, and which will also improve our quantitative capabilities moving forward. So finally, we do think about the environmental side of the world, and again, whether it's pesticides or wonderful PFASs, of which there are many, 60 plus, and some talks on that, persistent organic pollutants, which are everywhere. Our technologies are set up to do that. I'm just going to show you one example of a workflow and technologies we have developed to address these markets. So EFOX, which is Environmental and Food Organic Xenobiotics, so it's got a catchy name to it, is a workflow. Again, it's a workflow which was developed specifically to look at these environmental and food issues. So again, separations, this is done with a duo, so you can actually speed up your analysis, so we can do dual channels on this. Again, there's a productivity gain on that. And then we have two versions of the detector. One is based on an Exploris EFOX, which is really for profiling and screening. And then we have one versed on the Altis, which actually is more for targeted and regulated analysis. So this entire workflow can go both down two paths. One is screening, one is profiling, and one is really for doing targeted analysis. And again, the whole thing feeds into Chameleon, which now has new quantitative capabilities as well. So thinking about the hardware, thinking about the separations technology, and thinking about the software into one workflow is what we're trying to do. So I've covered a lot of territory there in a short time, but you'll see the technology and the innovations we're bringing, whether it's proteomics or structural analysis, or whether it's food safety. We're really trying to push the boundaries of science with our technologies. And also on the software side, we're embracing things like artificial intelligence, where to use those sensibly to make our instruments smarter, make our data analysis smarter. So this is a really exciting time. This is an exciting time for science, and I think for all of us, we're really excited about bringing these technologies to you, so you can actually push the boundaries. So with that, we've got a whole set of work streams now, and tremendous speakers who are going to talk around the different areas, and whether it's in proteomics or small molecules or food safety, biopharma, and these are world-class speakers, and I think you're going to enjoy the day in terms of the science we're presenting. A lot of it will be represented around these new technologies. A lot of them have access to these technologies, and they'll validate the things that we've just shown you today. So I want to thank you again. I always enjoy coming up here on the Sunday morning to show the new technology. I'm always proud and honored to be able to do that. And I really hope you enjoy the show and enjoy these breakouts. Thank you very much.